So today I'll talk about the ADP-Ribosylation factor 4, which is the ARF4 involvement in GABA homeostasis. Before starting, I'd like to thank to the Stony Brook Medicine Department of Pathology along with Stony Brook Cancer Center for helping us do all the experiments. And I also especially want to thank to Sam Chiapponi, who is a PhD student, who helped me out throughout the process of basically obtaining data and in the process of writing. The general disclaimer about this project is basically it is submitted and it's in the process of publishing. So all data have been already obtained by the authors and cannot be copied, presented, or disseminated by any other people because it contains information belonging to the research group, which may be confidential or privileged. So regarding to this situation, we expect your understanding. I'd like to give a brief information about the GABAergic homeostasis and what our ARF families are. So as you can see, the red indicates the glutamatergic projections. Also, there is the GABAergic projections, which are indicated in the green color. So the red is generally the glutamatergic, while the green is the GABAergic. So major glutamatergic projections arise from the frontal cortex, and in the frontal cortex, there is the involvement of anterior cingulate cortex, ACC, thalamus, ventral tegmental area, hippocampus, and the nucleus accumbens. Those neurons originate from the hippocampus and innervate into the hypothalamus, VTA, and PFC, and from amygdala to HE, HT, ACC, and NAC. In addition to that, on the other side, GABAergic projections, which are indicated in those green colors here, are widely distributed throughout the brain. So it is almost everywhere. And the major projections that are relevant are from generally the HT and to the occipital and parietal cortex. So the structural changes observed in the depressed patients are indicated in those boxes. So for the people who have depression, the prefrontal cortex has less volume while the glial cell density is decreased, while anterior cingulate cortex has the decreased volume and glial cell density, which is very similar to the prefrontal cortex. And the hippocampus also is decreased in terms of volume and glial cell density. All of those areas in the brain are generally showing same kind of phenotype where we see the glial density is being significantly reduced in the depressed patients. With this, I'd like to also talk about what are those ARF proteins that we were mainly focusing on for this project. So ARFs are small GTPases that play a critical role in neuronal trafficking and spine morphogenesis. Members of the ADP-ribosylation factors, the ARF family of small GTPases, and their regulators gained interest throughout the years because they are important regulators of the spine density and the morphology. There are basically three classes of the ARF families. Class 1 is ARF 1 to 3, while Class 2 is ARF 4, 5, and Class 3 is the ARF 6. Like other small GTPases, like RAB families, they act as a molecular switches that cycle between an active GTP-bound form as long as similar to the inactive form, which is the GDP bonded form. So when it is active, ARF 1 and 4 are very important for this early endosome creation and then in the trafficking of that early endosome through Golgi complex to the nucleus in all that trafficking. And when we look at ARF 1 and 4, which is our main interest is ARF 4 in the sense, is that it is highly important for the transportation through the COP1, which is literally early endosome content being transported to the nucleus. And other ARFs are basically important for different factors. ARF1 is also important for binding to PIP2, in the plasma membrane. ARF4 is also have been published to be involved in the cilia, and other ARFs like ARF6 are important for the focal adhesion molecules. So there have been two important studies that were indicating the GABA and the ARF interaction. One of them is published in 2020 in Molecular Brain by Kim H. and Jung H. This group have found that ARF6 is localized exactly on the GABAergic synapses. So what they did was generally taking cultured mouse cortical neurons from the mouse, which were in embryonic stage 15.5, and then they did this EGFB transduction to those cells and tried to understand if the signals were actually, if the protein was expressed exactly on the synapses of the GABAergic neurons that comes from the cortical area. And they found it was actually co-localized in the areas of the GABAergic synapses, which basically gave an important outlook because they then knock down or knock out those ARF6 proteins in those neurons in different isotypes of GABAergic neurons generally. And they found that its activity is indeed required for the GABAergic synapse development. As you can see in here, when there are the mutant forms of it, when there is the knockdown of ARF6, the development of the synapse is not being employed. When you compare with the control, it's significantly decreased. And they also did this analysis in vivo experiments. They also used a knockdown model, and they also had some mutant variants that would either the rescue mutants or the knockdown mutants along with the control group. And they also realized that when there's the ARF6 knockdown, the synapse development of the GABAergic neurons were not being developed properly. The other research that is actually very important for our research is that there is this paper that have been published in 2012, which we actually find it as a really reference article for what we are doing generally. They basically took the ARF4 and they made a heterozygous mice model. All of the mice were between 4 or 5 months of age. At the time of experimentation, they basically trapped the exon 1, and then they had this like loss of function mutants. And as you can see in their vectors, and when there's the shRNA, the knockdown mutants the knockdown mouse models were basically indeed knocked down. And they then further investigated if it was important for any type of neuronal development. They didn't find any difference in the motor activities. They didn't find any like anxiety behaviors in those animals. But which was really important is that they realized that the knockdown of ARF4 by the shRNA generally reduced the spine density in the mouse primary neurons. What you can see over here is literally significantly different when they have this like shRNA mutants, and you can see that when the ARF4 was then had to have a rescue model along with the shRNA, when the ARF4 was being rescued, then also the spine density different abnormalities were being rescued. So this was the first indication that it is highly important for the spine density. And then they made this ARF4 overexpression models, and they realized it indeed promotes the dendritic spine development in the primary mouse neurons. As you can see, when there was this overexpression models, they were actually healthy enough to grow healthy spine. Which basically led me to think, okay, so there were no motor skill differences or anything. Mice were not showing like massive differences. but ARF4 and other ARFs were mostly important in spine density, but also the GABAergic synapse development. That's why I wanted to first of all look at where it is expressed in the human brain, ARF4 generally. When you look at the brain, it is almost expressed in everywhere, which makes sense because it's a small GTPase, which is important for the COP1 through transportation of the early endosomes, and it is needed. But how about like where is it expressed very highly? And we realized that this is coming from the Human Atlas data. Cerebral cortex was one of the main areas that was increased. The was also the other area. And then I checked this reference paper, which was Lee et al., that is published in 2018, and they found that it was highly being expressed in oligodendrocytes and microglia macrophage interfaces. This immune system involvement, we are already very much well aware of the involvement of ARF family being upregulated on those cells, but this oligodendrocyte area was basically promising for us for doing further research. And then we wanted to check where it is expressed in the mouse brain, and we realized that it was mostly being expressed in the pituitary gland, but other than that, it was very similar to human. Hypothalamus was one of the main areas. In the cerebral cortex, it was high. Then again, the Lee et al. Also published where it is expressed in the mouse brain in terms of cellular level, and they were also indicating it was highly expressed in the oligodendrocytes. Then we started our project for back in 2020, and our main aim was not actually looking for a psychiatrist disorder or any type of neuronal development. Our first aim, because we are a skin lab, we work on the cutaneous biology, and what we wanted to look at if ARF4, all those GTPases were involved in the pigmentation defects, and that paper is already submitted and it's in the process of publication. What we first found was, yes, they were indeed important for the melanocyte pigmentation defects. We created a conditional knockout mouse because one of our collaborators, Greg Pazor, already had a universal knockout, global knockout, and the global knockout animals were basically unable to grow. It was lethal. So it is required to create a conditional knockout because as a lab we were working with the pigmentation problems. We created a tyrosinase animal model where we fluxed between the after the exon 1 and then after to exon 3. So the two exons were already like cleared out, and out of that animal model, because it was a conditional knockout, we induced the conditional knockout through the injections of tamoxifen intraperitoneal daily, and we harvested our animals at P66. And as you can see, which was basically after the anagen where the hair growth already happened for the second cycle. And as you can see that there was a coat color difference in the mutant animals, and it was promising.
I'm sorry, I had to stop here because I just learned that this pre-record presentation needs half an hour. Since we have very distinguished colleagues here and we didn't get a chance to, interact and ask more questions, it's not fair to, continue listening. So I would prefer close the session here and let the video run if you guys are really interested in listening. Do you agree with that? So with your permission, Mr. President, I would like to close this session. Please join me to thank Dr. Mitchell and Dr. Yuan to be with us and with their gorgeous scientific input into our Congress. So thank you so much for being with us. Siz lütfen videoyu oynatmaya devam edin.
Okay, you can keep on playing the video. If anyone is interested, they can listen to it.
But the further investigations for this experimental model actually led us to look for the neuronal development generally. But in order to be sure that the pigmentation defects were actually happening through the lack of melanocyte development, we did stainings of, this is coming from an RNA scope, so all the dots you see, not just the general green, but the dots are basically real signals. The melanocytes are DCT MITF positive, and as you can see in our mutants, there was almost like no double positives, which was significantly down, indicating indeed the melanocytes were not being properly differentiated. Okay. Then all of those situations led us to look further about how the shift happens. What we first found, because we had in vivo models and we wanted to look at what is really happening in terms of molecular level, we wanted to go back to the first initial developmental stage, which is where all the cells are coming from, the embryonic stem cells. Let's get the iPS, induced pluripotent stem cells, and then start the development, this differentiation from there in control cells, along with the mutant cells where we already did this cloning that we created shRNAs, ARF4 shRNAs, to knock down the ARF4 expression. We first of all plated our cells and then added ROCK inhibitor for them to become single cell level so we can actually see how the cells are changing the phenotype. And then did the transduction, meaning we knocked down the ARF4 and then started differentiation. As you can guess, in normal conditions when the ARF4 was there, with the melanocyte differentiation media, they indeed became melanocytic-like. The phenotype was actually showing that they were making the star-shaped phenotype, and then they were creating melanin pigments. But what was really, really important to us, that when the ARF4 is knocked down, it was indeed becoming something else, and it was looking like a neuronal phenotype where there were like this extreme, like longer dendrites. We didn't know about how the phenotype changes happen. So we indeed looked at if, first of all, the knockdown cells are creating any pigment, because they can actually be some sort of a malformed form of melanocytes, and they were not creating any pigments. And what is very surprising to us was that the cells that are neuronal are basically having this like cell body bigger than the other melanocytic cells. And what we've seen in the knockdown cells, it was indeed the cell body area that I just drew as spherical circle here. It was significantly higher, and we counted like thousands of cells. So what we have seen is they are indeed becoming something neuronal when there is the ARF4 knockdown exists, but it's looking like a neural crest-like, but there is only one way to be sure if they're actually staying on the neuronal crest. Why did the first option as neuronal crest came to our mind? Because in the developmental stage, even the melanocytes derive from the neural crest cells. So there are neural crest cells that can actually become neuronal cells, or in the presence of Wnt, they can actually endorse to become a melanocyte. So this was the main aim, understanding that ARF4 is actually one of the major elements that makes that linear, like that developmental shift between the neuronal cells or the pigment cells. We indeed found that knockdown of ARF4 in differentiating iPS shifted the differentiation of melanocytes into a neural crest-like. Before showing this like individual gene expression analysis, which comes from the qPCRs, I'd like to give a brief information. So this is already published data. The iPS actually become neural crest cells in the presence of Wnt, BMP, and TGF-beta. When I say BMP, it doesn't mean that it's overactivated. There are elements that are involved in this differentiation. And from neural crest, if there is BMP4, EDM3, cyclic AMP in the present, they become melanocytes. If there is neural growth factor, BDNF, all those, they become neuronal cells. Now we're actually suggesting that ARF4 is actually important and its presence is needed for the melanocyte differentiation, but in the deprivation of ARF4 actually endorses neural crest cells to become a neural cell. First of all, we wanted to check if our shRNAs worked, and as you can see, the knockdowns work properly. And with the differentiation, the ARF4 is trying to be rescued internally by the cell along with producing other ARFs to compensate its job. But what was really important is the melanocyte markers. When there's ARF4 knockdown, melanocyte differentiation markers were significantly getting down, while the neuronal markers are significantly getting higher. Which was very surprising and very interesting. And we further then looked at neural crest markers, and we realized that indeed the knockdown of ARF4 immediately decreases the neuronal cell markers, but then with differentiation and the lack of ARF4 was endorsing it to be increased, and they were endorsing the neuronal cell differentiation. But as molecular biologists, to be fully sure that they are neural crest cells, the cells of neural crest generally it is published to have the ability in the presence of matrigel, which is basically a three-dimensional molecule, three-dimensional extracellular matrix material that will help cells to make this three-dimensional neural tube-like structures in cell culture systems. So we had our control cells and the mutants that had the ARF4 knockdown, and then also the control cells are basically treated with the differentiation media. As you can see, they couldn't form any type of neural-like tubes. And when there was the knockdown and the differentiation, they were indeed making neural crest cells aggregating and making a neural tube structure, which was really fascinating, and that was a proof that indeed the cells were becoming neural crest-like. Now, I'm now halfway in my presentation, and I'd like to give a brief information about this physiological and pathological human neurogenesis. This is basically a developing neural tube. After we've seen this neural tube development, so in vitro environment, the neural induction from the pluripotent stem cells to the neural tube is being employed through different activations of different pathways. First of all, for cells to become smooth muscle cartilage, Schwann cell, peripheral neurons, etc., they literally need the dual SMAD and Wnt activation. For them to become rosette neural precursors and cortical neurons, they also need the presence of retinoids. Which is very important for us, is that for them to become GABA neurons, there should be a dual SMAD activation and Wnt inhibition. So this is basically a proof if there is the Wnt inhibition happens properly, because the presence of Wnt is actually endorsing cells to become melanocytic. The lack of Wnt causes them to become neuronal-like. So since we have found the lack of ARF4 also showing some kind of a general phenotype, what if there is a real Wnt inhibition in protein level through the lack of ARF4? To be able to further prove that, we needed to be sure in our in vivo models, plus in our in vitro systems, the knockdown of ARF4 was indeed causing Wnt inhibition. We first wanted to look at the ARF4 flox tyrosine cream mice, and let me just give a brief information about where the ARF4 might be involved in the Wnt pathway. When the LRP6, which is basically the receptor, Wnt ligand binds to the LRP6 receptor, there is the Shevel protein that comes and binds to the receptor, where they probably activate the ARF4, which turns ARF4 GDP form into the ARF4 GTP form. And in here, there are some inhibitors that might be involved, but when the ARF4 GTP form actually assembles, they make the production of PIPs to become PIP2. And when I say PIP2, it's phosphatidylinositol 4,5-P2. And when this complex assembles, it gives the 2-phosphate here to bind to the LRP6 receptor, which then further activates the whole Wnt pathway complex to happen. If the Wnt pathway is active, the complex basically translocates that binding complex endorses beta-catenin to translocate from cytoplasm to the nucleus. If it stays in the cytoplasm, it means that it's going to get degraded, so the Wnt pathway is not active. So to be fully sure about it, we had our hair follicles that we had in our in our previous model system, and we took a non-pigmented, which is mostly the mutant, and the pigmented control animals. And we did double staining. TRP2 is a melanocytic marker, and we were looking at the double positivity of those cells if the melanocytic cells were indeed expressing beta-catenin, which indicates that there is an active Wnt. And we realized that in our knockout animals, the double positive, the nuclear intensity of the double positive cells, which indicates the active Wnt, were significantly lower, which literally suggests that it was downregulated. Wnt was downregulated in our mutant animals. We further looked in vitro, and we took some low pigmented melanocytic cells and then did siRNA knockdown, did no media change, and then did RNA and protein extraction. First of all, we checked if our ARF4 knockdown worked with the siRNAs. As you can see, significantly lower levels of ARF4, which indicated that the knockdown worked. Beta-actin was stable as in housekeeping, and then we checked the downstream elements of the Wnt pathway. And we realized the overall beta-catenin was okay, it was stable, because this doesn't actually give any information about the nuclear or cytoplasmic localization of beta-catenin. The overall was significantly lower, but generally same. Which was very important is that the overall LRP6 is the same while the phospho-LRP6 was gone, which is a huge indicator of active Wnt pathway in our mutant animals. So, which basically led us back to the idea that this phosphate addition to the LRP6 is not being employed when the ARF4 is not in the present. I mean, the active form of ARF4 is not in the present. We further then did relative mRNA expression analysis by qPCR, and we realized that most of the downstream pathway elements were significantly downregulated, and we further wanted to have a rescue model of the system, and we took the same cells, again did ARF4 knockdown, and did starvation and added Wnt3A ligand, which would, in normal conditions, would activate the Wnt pathway. And we realized even in the active addition of Wnt3A, when there was ARF4 knockdown, cells were not actually activating the Wnt pathway, and the nuclear beta-catenin intensity was not being rescued. This is basically the end of my presentation. This process is still being investigated. We are believing, of course, there are like further more important findings through the melanocytic side of the project, which was already submitted and about to be published soon. While this neuronal part of it is also more fascinating in terms of understanding how this ARF4 is like mostly involved in this like phenotypic shift. That's why I'm not actually showing you any like conclusion slide, because we're still in the process of brainstorming on it and doing further investigations. We will be happy to collaborate if you have any opinions. So you can literally reach me through my email address for your questions or your comments. I'd like to especially thank to Dr. Jiang Chen, who was basically the PI through this project of the melanocytic side, and Greg Kazor for doing the RF work in his laboratory that would literally supplementarily strengthen our hypothesis. Adam Nackley, who's the med student that helped me throughout the process, the Department of Pathology, and the head of the Department of Pathology, Professor Kenneth Schroyer. This project's general Funding is coming from the NIH, so we also would like to thank to the NIH. Thank you for listening, and I will be happy to answer your questions either directly to me or through my email.